Thank you so much for the introduction. Welcome again to my talk. And then I hope you are already enjoying the conference and then the lunch. I think it was pretty amazing conference so far. And it was also my first time in Prague. And yeah, today I'm going to talk about how to build an open source machine learning platform in biology. So let's get started. Who am I? So I'm Furkan, and I'm from Turkey, living in Munich, and also work there. And then this is the region where I grew up. Most probably you see from some advertisements, and of course I love hiking in Munich. And my background is molecular biology and genetics. And now I work as researcher and data scientist in a biotech, and then I'm combating against the cancer. And previously, I worked as computational biologist and data scientist, all in research area. And at the same time, I'm a community builder, and I'm expanding the Bits in Bio chapter in Munich, which is aiming to bring all the bioinformaticians to the one place to exchange the idea and network. So that's what I am. And here is my agenda. At the first glance, at the first section, I will try to make a bridge between the biology world and also the computer world. And then I'm pretty sure that there are some people who are skeptic about the biology part. Please don't be stressed if you know what the DNA is. So we are fine. And then I will talk about the machine learning in biology. And there are some pitfalls and issues. And then I will discuss all of them, and then later I will introduce our platform, omics, and then hopefully I will do demonstration, and then we'll be getting your questions. So let's get started with biology and data. Again, no stress about, so stay with me. So right now we are in the cell scale. So you can consider the cell as the computing unit. So this is where all the complexity going on. And in molecular biology, we have a phenomenon called central dogma. Central dogma dictates how the genetic information flows. It's one direction, and everything starts with the DNA. You know, it's like an alphabet of 4 letters, A, T, G, C. And then it replicates itself, and in this case, it's your Python file, so which is your recipe. And then we transcribe this DNA to the RNA, which is your interpreter in programming world. And then finally, we have the proteins, which makes the actual function in your body, and which is your executable program or the final script who's doing the actual work in your computer. And if you collect all of those codes, which refers to the genome, and the same applies for RNA, which is transcriptome. If you collect all protein library, it's called proteome. So that was it. It looks simple, but I would like to give one bonus point, which is CRISPR. Why I'm giving this? Because it looks very simple, and then here I would like to give one example. In your favorite IDE, VS Code or PyCharm, whatever, there is a functionality, right? Find and replace. Okay, it's not sexy, it's not a word to talk about, but having the same functionality, meaning that finding something and changing within our genome ends up in a Nobel Prize just 3 years ago. This is how the complicated the things are within our cell. So far, we were in the cell scale. Now, let's zoom out a bit and let's talk about the samples. In biology, we work with the samples. So it could be blood. I'm pretty sure you already sent some blood, maybe to some ancestry test, or maybe your saliva. And there are also some other types of sample, and each sample type has separate preparation for the following methods, but I will not get into any details of it. And there's the most important section of this transition. Literally, this is the phase where your biological sample is translated into the bytes. So there are very sophisticated and very expensive machines for translating those biological samples into a quantified version. And finally, then everything goes in that direction, and from bases to bytes, At the end, there's a discipline came up called omics, which is aiming to analyze the biological data sets. As you see on the right, there are several layers. If you are talking about your DNA and genome, there's a genomics for proteins, proteomics, which is the field I'm working in. But the ultimate goal of the whole field is multi-data integration. So we would like to also know about your genome and proteins, and also your phenotypes and all the things about you. So, and then I would like to share some few milestones about how was the journey so far. And I think this is my personal opinion. The Pandora's box was opened early 1990, and because it was like decided to take the sequence to human genome, which means that we would like to know what is the ATGC order in a human being. And it took us 13 years to finish this project with $3 billion of cost together with thousands of researchers. And then we finalized this project in 2003, but it was not end. Just early last year, we finally I have the first complete version of our genome. So we tried to keep fill some gaps. There are still gaps. So this is how the complicated in our area. And the ultimate goal is to reducing the cost for the human genome sequencing. It started from $100 million only for one person. Now it's already up like around 600. And ultimate goal is to make it like 100 and democratize for everyone. And of course, let's share some more data about the biological data itself. And I took this statistics, which is recently published. This is the last report from the EBI. EBI is one of the largest consortium in bioinformatics field. And in this graph, you see the daily requests for the database service and the other services for the EBI. And as you see, there's a huge trend because the data is growing over there, and there are a lot of people are also willing to put their data to there. So, which makes an open science community, and which is good for us. And in terms of the volume, if you are wondering, currently for the EBI, they already reach out to 75 petabytes in total. It includes image data, genome data, proteome data, and several other types. And in yellow line, as you see, there is an increasing trend, and we are sending more data and more. Of course, when we talk about the growing data and in volume and also in the complexity, machine learning always comes into the scene. And I'm pretty sure that you already heard a lot of applications of machine learning in biology, especially during COVID times. You see there was a lot of the machine learning models, whether they're trying to predict whether you have COVID or not, based on your tomography scans. And there are, of course, several other ways based on the category of your data. But today I'm going to talk about the tabular format, because this is the field where I am in, and we are working with the tabular data. Of course, the ultimate goal of those applications are helping the medical doctors diagnose the cancer earlier and also discover new drugs that works for human. So at the end, we are building a platform, but let's start with what are we working with? What's this tabular data looking like? And here, we are gathering a lot of samples from Alzheimer patients and non-Alzheimer patients. The sample type is a cerebral spinal fluid, which is taken from your spinal cord. And then we are having each row represents two patients, and we have the proteins as features, so which means that how much protein is expressed over there. And of course, we collaborate with the hospitals, and then we get more data, and of course, we have the target. So I answered the what. And now, why we developed this platform, right? The first reason is identify some data patterns, so then we can diagnose the disease earlier, so then we can save much more life, and provide also the support for medical doctors. And one example is the BRCA. Maybe you heard Jennifer Lopez got diagnosed with the breast cancer thanks to those genes. So that was one of the example for how we identify the patterns and diagnose. And for misleading metrics, unfortunately, this is so much tragedy about academia. I'm not blaming anyone here, but this is the case. For instance, if you consider this case, if you have 100 samples, 90 of them are like healthy and 10 only in rare disease patients. But if your model predicts everyone is like healthy, you will get 90% accuracy and 0% of F1 score. And guess what? Which is the most reported metric unfortunately, in academic papers, and they are getting accepted. And of course, the machine learning pitfalls. Here, I would like to go over some top 3 machine learning pitfalls. I'm not sure whether you attended the keynote speech yesterday. Evelina gave a really nice a base for me, like a reproducibility issue in the science. Unfortunately, the same thing exists also in machine learning in biology. Unfortunately, there's a lack of transparency in the methods, and as you see in one example from a published paper, accepted and published paper, so there are some methods, but who knows how to get that method in that way, because there are several ways to tune the parameters and get the model working. The other one is data and target leakages. So I found this amazing literature review which collects several papers in the field, and see the pitfalls. There are no trained test split, there are some other leakages, which is complete mess. And again, they are all accepted and published papers, so we have serious trouble in academia. And last but not least, some methods include stochasticity, which means the things happening in a random way. So you have to provide an environment where another people or researcher can reproduce. But unfortunately, again, another proof that I'm not lying. So that people are just randomly select the data, but there is no way to reproduce for us. So what's wrong in the ML, in biology? A lot of things, but the main issue is unreproducibility. So in the conference where a lot of software programmer, I know you know, it works on my machine. So we have as the same, hey, it's reproducible on my machine, but unfortunately we can't send your machine. So I talk about what and why, and now let's talk about how. Here we're gonna talk about all Python. So after listing all of the needs, we just came up with a list, what to do. So, and it should be, first of all, open source, then everybody can see, and should be reproducible, and meets the needs of the biologists in a way that it also follows machine learning best practice. So there is no leakage, no bad application. So here is my architecture for omics-learn. We develop omics-learn. You can find on omics-learn.org, and here is our architecture. In our platform, we have 6 components. Of course, we are very lucky that in Python ecosystem, there is a really gold standard tools, and one of them like a data science stack, like a pandas, numpy. Of course, we use them for data analysis. And for making the lives easier for biologists, we use Streamlit. I will come to the reasons why we use Streamlit in the following slides. And same for XGBoost and Plotly. And for machine learning, we use the scikit-learn. Again, it's like a gold standard. And Plotly for visualization. And another important thing was the test, because we would like to ensure the results that are coming out from our tool is reproducible. We made several integration tests, and then we are running on GitHub Actions so that in every release we are having the same results again and again. This was one of the key issues. And of course, we prepare a lot of documentation on Sphinx and connected to read the docs, so you can find everything on omics-learn.org. Finally, last but not least, we realized also, because I'm working closely with the biologists, also I'm a biologist, and the people don't want to spend some time on Conda installation, some other Python stuff, because they don't have time, literally. And what they want also, we already hosted a web service, but another problem is they always don't want to, most of the time, they also don't want to upload their data to a web server because of confidential data or sensitive data. So we have to bring this software to the scientist or researcher in a good and easy way. Therefore, we use PyInstaller. I was happy with that. So we made a couple of clicks. You make a couple of clicks, and then you install the whole stuff very easily in minutes. And this is how we structure our repository in case you would like to build your own platform in your field. I just would like to show how we organize the things. And I would like to also highlight for the deployment. So for deployment, we were using AWS on our sites, but then there was really something cool happened, and Streamlit announced the community cloud, and then we can easily synchronize our master branch with the Streamlit cloud, and then it's automatically build the code and then deploy it. So that's what I can also recommend to you. Then let's talk about tech stack. Of course, you pick your tech stack based on your time and also look at skills that your people have. For our case, we would like to have the rapid prototypes. And therefore, we didn't want to go a native solution. And therefore, we picked Streamlit. The reason why I'm saying rapid prototyping is, as you see in the screenshot, you can just import the package and just make this, write the multi-select and this fancy, also looking good environment. And then you don't need also any callback to save those, what the user picks. So if you decide, like, if you point this to a variable, it will be saved without any callback. That's one of the biggest thing, because I don't want to deal with the hundreds of callbacks. And there is no HTML, SSS, and Python mix like in the other packages like Dash. And this was really cool for us. It's just pure Python. And of course, there's a free deployment, as I mentioned in the last slide, and it works very well all the data science stack like Pandas, NumPy, Plotly. Of course, everything comes with pros and cons, and it's not that much flexible, of course, like a Django or Flask or on your own, but this is the issue with the rapid prototyping, because if you want to rapid, you have to give some disadvantages for flexibility. And performance, I put question mark because for us, and we never experienced, hey, this is slow, we never heard this, but of course it's not as speedy as the Django or like a native solutions. And the testing, unfortunately, I used Selenium, and today I would like my attendee to live longer, have better eyesight, please don't try. So it's like, I want you to live longer. So for the XGBoost, actually the biological data is really complex and it doesn't follow a linear, like a linear pattern. So we have to integrate some decision tree models. And if you look at the Kaggle competitions and other places, also papers, you will see that the XGBoost will, like always, this package you will be seeing. And therefore we try to pick that one. And last need the Plotly. Why we picked the Plotly is publication-ready plots. We definitely know that researchers would like to take the plot, just click the download button and put to their papers, because they don't have time and they really don't want to manipulate the access and make it like a beautiful in Adobe Illustrator or whatever other programs. They just want to download and paste it into the paper. And therefore, we need a lot of customization and also interactivity. Therefore, I can suggest also you to use Plotly. Finally, this is the highlights. So Streamlit picked us App of the Month. At that time, I think it was March 2021. And then we published our paper in an open access way, so which means that you don't need to subscribe. We paid for this. So you don't need to subscribe or get any other do any other things. You can just visit, and then we got almost like a 4K in the last 6 months. And then the journal also decided to put us on the cover. So this was also really nice for us. And then, yeah, that was the highlights. But this is the slide where I'm most proud of. We have happy researchers. So what it means is, so several, even though it's published just 1 year ago, we have a lot of citations, even like one came just 5 days ago. And we have really cool analysis that use the omics-learn, and then they found some really cool genes that doesn't appear in normal statistical approach. And then they found some other stuff, and they also realized also it's overlapping in the normal statistical approach, which means that we don't provide some bullshit results. Let's be fair. And also other the papers, and more is on coming. And let's go for demo. Yeah. So there are two lanes in our platform. One is the main window on the right side. On the left, should I zoom in? Can you see from the back? If you give thumbs up, okay. I see thumbs up. Okay, thanks so much. So on the left-hand side, so let's start with the right, sorry. You can upload any data, like most of the time you work with the Excel or CSV or TSV, you can just upload, and it's like pretty easy, and then you will visualize the data, actually inspect the data, and there are several data handling manipulations that just happen in the platform, so you don't need to go any Excel or any other tool to filter something. We already provide this to you in our platform. So for the classification target, in this case, this is the Alzheimer data. We have the clinical results, and then I provided already as positive as the Alzheimer, negative as the non-AD. And on the left-hand side, you see the configurations. So if you don't know what to click, what to do, there is like a hyperlink for all sections. And for this, we put actually a lot of efforts for documentation, and then you can see several explanations, what's this and what is what, and how can you utilize that functionality in our platform in easy to understand, in an understandable way. And you can also pick some imputation methods and choose also the several classification algorithm. And for the simplicity and also the sake of the time, I will just run the results, and then at the end, you will get the results. Yeah, this is actually the researchers, what researchers use the most, because those are the proteins ranked by the importance, which means that this protein could be the best candidate for diagnosing the person. In this case, if we check this protein, what it is in the search database, it was a tau protein, which is already known for the Alzheimer. So it also shows up in our platform. So, which again, we prove that our platform finds good biomarker candidates. In this case, you can consider those proteins or genes as biomarkers. And you can have the several plots, ROC curve, PR curve, and confusion matrix, and all of the other metrics. I will not go into the detail for each, but two most important things are the summary. As said, the reproducibility is the key issue. Therefore, we thought it would be nice if we provide such a summary text for researchers. They can just download and put into their paper, and then everything is like a relax, and then they can reproduce all the analysis results in our platform. Not only that, we also provide the session history. If you remember my screenshot from the Streamlit, I put every variables into a dictionary. The reason to reach out everything at the end. The thing is that you can see what is clicked over your sessions, over all the page during your session, and then you can also download it, provide to your colleague or provide to the journal, and then everything is definitely reproducible. So that was overall the demo, and let's go back to my presentation. And yeah, and Dikimoch, thanks so much. And yeah, you can contact with me on Twitter, LinkedIn, or you can just scan me and just go my website. And yeah, thanks so much for joining me and listening me.
Furkan, thank you so much.
Yep.
I can see the hall is now full as everybody's back from the snack.
Yeah.
I'm happy about that. I do have questions for you here, so I will start right over. Vojta asks, I assume one instance is for one user, or did you think about multiple users working on the same instance, e.g. to save the load time for large models?
You mean the working at the same on the if the who has the question if you okay, so do you mean like working on the same time, I mean the real time? We actually didn't think about that, and I think it's not will be feasible for the Streamlit. Yeah, I'm sorry for that.
Okay, thank you for the answer. If you did this again, what do you wish you knew?
In terms of the result or the whole process of developing? General.
Yeah, in general.
Actually, the ending up with a journal took us 1 more year. So we had a really serious issue, even though like the field itself really needs this tool. We are having so much difficult time to get our paper accepted, but at the end it's getting accepted. What I would like to try is not too much wait for this, just because we already published on the preprint, and then I wish I would put more effort to functionality-wise and also get much more valuable feedback from the reviewers. But in academia, sometimes it's not going in the perfect way.
Limitations, I see. Scaling question by Vojta. You mentioned you're using AWS for hosting. Does this mean you can scale easily to many users in the future?
We were using the AWS in the past. Now we switched to the Streamlit. I think they are taking care of the scalability. So we just would like to avoid this operations on our own and then switch, because we also paying the cost. And yeah, you have to also find the way to maintain this work because it's an open source and you don't pay anything. So for the AWS cost, we would like to avoid and then switch to the Streamlit. So therefore, I can't answer right now on that question.
And the last question, do you have any open source contributors?
This is like a bit disappointing for me. So we were only 2, me and my ex-team leader so far. But we have a lot of researchers who use, but not developers. Maybe one of you can help me.
If you're looking for a project?
Yeah, just find me.
Perfect. Are you going to be on the after party?
Yes, I will be.
Perfect. Thank you so much.
Thanks so much, and yeah.
Thank you to everyone. One more thank you.